guess you're all here, so you probably didn't enjoy it all that much. Sorry, I couldn't make it last night. Okay, are there any initial questions that were raised from yesterday that you would like to have addressed before we start again? No, really? Okay, I know there was at least one.
Okay, quick question. Does FreeSurfer use NU correct to the intensity correction?
Does FreeSurfer use NU correct? Yes, it does.
Does it differentiate between 1.5T and 3T scanners and make a correction for the spline at different distance?
Does it make a distinction between 1.5T and 3T scanners?
Yeah.
We do have something in Recon All that allows you to specify that it's either a 3T or a 1.5T, and I think that will do something to do correct. I don't know if it will do anything to the particular parameter that you're talking about.
So it doesn't pull it out of the DICOM automatically?
It does not pull it out of the DICOM automatically.
Okay, so is it optimized then for a 1.5T?
I think it's probably optimized for 1.5T. But that's not the only thing that we use for bias correction. So there's we basically have a 2 or 3 step bias correction algorithm that's working in the background there. Anything else? Okay, so this morning and today we'll move on from the individual subject that you were working with yesterday. So yesterday you learned all about how you take a T1 image where gray matter is dark and bright and white matter is bright, and run that through the Recon All processing stream. So that goes through the volume-based stream where it does the intensity normalization, skull stripping, volume-based labeling, and then white matter extraction. And then from that extraction, after it extracts it, it then extracts the surface based upon the surface of the white matter. And the surface is a list of vertices, their nearest neighbors, and an XYZ coordinate for each vertex. And so with that information, you can compute things like area. Once you have the wide peel surface, you can compute thickness. You can compute distances along the surface. And then we can use that to compute curvature as well. And based on curvature, we can perform this intersubject registration and map ROIs back into the original space. So yesterday you looked at Recon All, which runs the individual subject, and of course probably what you're going to be interested in more is doing some sort of group analysis, and that's mostly what we're going to be talking about today. So this talk is on surface-based analysis, intersubject registration, and smoothing, which you can basically think as pre-processing steps after you run Recon All, but before you go on to your surface-based group analysis. You run through these two things. And then Eugenio is going to talk about group analysis. He'll do a tutorial, and then he'll talk about correction for multiple comparisons on the surface. And then I think you do another tutorial, and I think Martin is going to talk about longitudinal analysis. And so that's the whole day today. So first I'll talk a little bit about exploratory spatial analysis and what that means and why you would want to do it. And then go through the coordinate systems, 3D, which you're probably used to if you're used to doing volume-based analysis, or 2D, which is the surface-based analysis. And then I'll go through intersubject registration in the volume and on the surface. And then I'll talk about surface-based smoothing and surface-based clustering. So an exploratory spatial analysis is something that you do when you don't know where the effect is going to be versus a region of interest analysis where you'd say, I know I'm looking at the campus and I'm only going to look in there. If you don't know where the effect is going to be, then you have to do this exploratory spatial analysis, which means that you have to analyze each voxel or each vertex separately. And so what that means is you analyze each one separately, and then you create a map out of that, and then you correct that map for multiple comparisons, which is usually based by doing some sort of cluster analysis. So let's say that you're interested in the effects of age on cortical thickness. So this is from a study that was done by David Salat almost 10 years ago now. And this is also going to be the study that you're going to be working on in your tutorial as an aging study. So this is something that Bruce showed the other day, yesterday, showing these are maps of cortical thickness ranging from 1 millimeter for dark red or 3 millimeters for bright yellow. And you can see that the 18-year-olds have nice, juicy, plump cortices. It's all nice and yellow and everything. And as you get to 88-year-olds, you see a lot more red in there, meaning that cortex is thinning. And so, of course, we're interested in what areas are thinning and what areas are not. And so, one way you can do that is to do an actual study where you compute statistics. So this is a 4x10 matrix of 40 different subjects. So this is the thickness maps that you were looking at yesterday, mapped to our FreeSurfer FS average, which is our common coordinate system in surface-based space, kind of like a Tallarat of surfaces. And so all of these are in vertex for vertex alignment. And so if I selected one particular vertex, I can make a little scatter plot here. The horizontal axis is age, and the vertical axis is thickness. And then I can just fit a straight line to those two, and you can see that the line is going down. The slope is negative, meaning that things are thinning with age. And I can do that for every voxel. I can compute a p-value for that. And based upon that p-value, I can create this statistical map showing places that are p less than 0.01 that are changing with age. And blue in this case means that there's a negative age correlation, and red means that there's positive age correlation. You can see that there are a lot of places in the brain that are negatively affected by age. Here's an exploratory analysis with FMRI, where we took the FMRI time signals and we mapped them onto the surface and smoothed them on the surface, and then performed the fMRI analysis looking at, in this case, this is a words versus fixation paradigm. And so you can see here on the left hemisphere, activation of Broca's area. This is probably some sort of motor area. You see visual cortex lighting up back here. The outlines here show you the APARC, the desiccant atlas. And you can see that the blobs don't necessarily line up with those regions of interest. So if I've done a region of interest analysis, just simply averaging inside of these anatomically defined regions, I might very well have lost all of this activation and would have concluded that there had been no response with respect to the brain. So exploratory spatial analysis generally requires that you smooth your data. And I'll talk about exactly what smoothing is in a few slides. And you do that to increase the SNR because at a given voxel, the noise is generally fairly high. The signal to noise is pretty low. So you smooth in order to boost the signal to noise ratio and get more confidence in what your results are. Also, for group analysis, for group spatial analysis, you have to have the subject's brains aligned in a common space so that you can do a voxel by voxel analysis. So neither spatial smoothing nor intersubject registration are needed for ROI analysis, which is why people really like to do ROI analysis. And smoothing and intersubject registration can be performed in the volume, or they can be performed in the surface. And of course, what we're going to be arguing is that doing it on the surface is better than doing it in the volume. Why that is related to what the surface tells you. So this is an image from Marty Cerino's now classic 1995 paper in Science. I believe FreeSurfer has been around now for 20 years. Marty was one of the original programmers of FreeSurfer, and he was primarily interested in the organization of visual cortex. And so what he did was, I think this is actually his brain, where he subjected himself to an fMRI experiment designed to study visual cortex, in which he had two stimuli. One was this expanding ring that was periodic, going out like this, and the other was like a hand of a clock that was going around like that. And based upon these two stimuli, he was able to map out where the different stages of visual cortex are. So there's V1, V2, V3, and V4, and so on. And when you look at this on a flat map, again, this is Marty Cerino's cortex flattened. The bright gray is our gyri, dark gray are sulci. And these patches here, these stripes, are the different stages of visual cortex as described by the fMRI analysis. And so you can see that on the flat map, these things are extremely well organized. You got V1 as this bright yellow, V2 as blue, V3 as yellow again, and so forth. And when you look at it on the flat map, the organization is just perfectly clear. However, when you look at it in the volume, you can see that there's almost no organization to it at all. And so this is one of the primary reasons why you would want to use a surface-based analysis, is that the cortex is defined by nature as a two-dimensional structure. And so what has to happen, so here's another two-dimensional structure here, right? This is a paper bag, all fabricated from a single flat sheet of paper. But this kind of thing would not fit into your head. So what has to happen is that the fold is spin up like that in order to fit it inside a head. But it's still a two-dimensional structure. And so it can still be unfolded. And so when you unfold it, you still have creases that are on it that indicate what the folding pattern is. And so that folding pattern is important in the human brain because function tends to follow the folding patterns, as indicated by Marty's analysis here. So V1 tends to be in the calcarine sulcus, V2 along the banks of the gyrus, V3 and V4 on the next banks of the gyrus. And so function tends to follow the folding patterns. And so it's best to analyze the data using the surface-based analysis, because then you're in the coordinate system, kind of that nature really intended the brain to be analyzed in. So what I'll do now is go through a volume coordinate system, the surface coordinate system, and then the volume smoothing, and then the surface smoothing. So what do you mean by coordinate system? It just means an XYZ coordinates, where in this case, for a volumetric 3D-dimensional coordinate system, by definition, you have three different axes. One of the axes is going to come out of your ear, that's usually the x-axis. Another is going to come out of your nose, that's the y-axis. And then another come out of the top of your head, and that's the z-axis. The axis is defined by really two things: the directions of the x, y, and z, and the origin. And so in a typical volume-based coordinate system, that could be something like the Colorado slice. So those would be the imaging coordinates. Or you could have something like a Talairac coordinate system, where you're defining the origin to be at the AC-PC line or at the AC. And then at each XYZ, you have some intensity value. And that's how you define where, what the brain is, and how you create, pretty pictures like the one that I have here. So that's just what a three-dimensional coordinate system is, just an XYZ and origin. On the surface, it's actually exactly the same thing, except that it's in 2 dimensions instead of 3 dimensions. So in this case, on the left side, what I've shown is a flattened map. The green are the gyri, and the red are the sulci. And so you can imagine it's just like a sheet of paper, where it's just you have the x-coordinate and a y-coordinate. But the definition relative to like what your head of those coordinate systems isn't entirely clear. And in reality, we don't use this sheet of paper type of coordinate system. We use a different 2-dimensional coordinate system, which are shown here on the right, which are latitude and longitude. So this is how you would locate something on the surface of the Earth. So the surface of the Earth is a 2-dimensional structure, just like the brain. And so you can locate a given point on the surface of the Earth using latitude and longitude. So this whole surface is continuous. There are no cuts in it. And so that's one of the reasons why we represent each hemisphere separately as a separate surface. We can also have a value at each point, for example, thickness, or in this case, I have a curvature that's mapped to this spherical coordinate system. And the way that we get to the spheres is that we just take the inflated and we keep on inflating it until it's very spherical. So using these coordinate systems, we can do intersubject registration. So the way this works volumetrically is to basically take two brains, and you can imagine like putting them like on two pieces of paper almost, and shifting them up and down and moving them back and forth and rotating them. And in addition to, you can stretch them in various ways. And the basic idea is that you're going to try to match the intensities on a voxel by voxel basis. So if it's in a linear transform or in a fine transform, then you have 12 degrees of freedom, because you can translate your head in three ways. So you can move your head back and forth like this. You can move your head back and forth like this. Move your head up and down like this. And then you can rotate your head in three different ways. So you can nod your head yes. You can shake your head no. No. Maybe. So those are the 6 rigid ways that you can move your head. And so what the intersubject registration tries to do is to manipulate those 6 parameters plus 6 more. So you can stretch your head left and right. I can't do that. Back and forth, up and down, and then you can shear it, which is like taking a square into like a trapezoid kind of thing. So those are the 12 ways that you can manipulate the image to try to get one image to look exactly like the other image, like voxel by voxel intensity. And there are also ways to do it in a non-linear way. But the linear way is pretty bad because, like is shown here, the red blue lines are the surfaces from the first subject superimposed on the second subject. You can see there are a lot of places where you just don't get very good alignment of cortex, especially. It does actually a fairly good job for some vertical areas. So surface-based is actually, again, okay.
Are you using one of your own subjects as the template image, or is this about aligning to a standard like CalRX space?
So the question is, are we using our own subject, like a subject from your study as a template image, or are you doing it to some atlas that we supply? So we supply an atlas, and I would say 99.9% of FreeSurfer analysis is done by registering to that atlas. There are a few people who will go and create their own atlases. I've done it a couple of times and didn't notice a big difference. But of course, your mileage may vary. But most of the time, people are using the FreeSurfer supplied atlas. So in 2 dimensions, things are actually really a lot easier because you really are dealing with sheets of paper at this point that have a pattern on them. And you can imagine like putting the pattern, like pulling the sheets up to the light and kind of doing this and rotating it and trying to get them to line up. There are a couple of important differences. One is instead of using like the MR brightness or intensity, we're using the actual curvature folding patterns. So this is where doing the surface-based analysis really has the advantage over doing a volume-based analysis, because we truly are aligning the anatomy. So in this way, we can make sure to get the folding patterns, like V1 aligned with V1, and superior temporal gyrus to superior temporal gyrus, and superior frontal gyrus to superior frontal gyrus. And in reality, we don't use these sheets of paper. What we use is the sphere. And as the analogy I made yesterday, you can imagine having, each person's hemisphere is like a planet. And this planet has mountains, which are gyri, and valleys, which are sulci. And And so you have major continents, and you have oceans, and you have small little mountains and little hills. And so you can imagine like putting these two planets together and trying to rotate them around to try to get the mountains aligning with mountains and the valleys aligning with the valleys. It's a nonlinear transform. So it allows for local stretching and compression. This nonlinear stretching and compression is sometimes called a morph. And since we do it on the sphere, you'll sometimes hear FreeSurfer people refer to this as the spherical morph. If you ever hear that, that simply means that this is just the surface-based intersubject registration. So this allows us to create a surface-based coordinate system. So, and the way this works is, as I mentioned before, we generate an atlas that's based upon 40 subjects. And these are aligned, so we aligned 39 subjects to one subject, and then created an atlas after that from 40 subjects, and then realigned everybody to those 40 subjects, and then did the whole process over again. So after 3 or 4 iterations of that, you pretty much remove the fact that you created your initial atlas from an individual subject. So this is what we call FS average. So FS average has a subject folder, just as if Recon All had been run on it. So you remember you ran Recon All yesterday and you created a subject folder for BERT, and inside of that was your MRI folder, and inside of that you have the orig.mgz and your T1.mgz and brainmass.mgz and apart plus asec.mgz, and in the surface folder you had all these different surfaces, LH orig, LH y, RH peel. So this FS average subject has all of those things in it. It looks like somebody just took it and ran Recon All. But in reality, it was created through a separate process to make it look like it's an individual subject. And so when you analyze your group data and you project it onto the surface and look at it in the volume, you'll bring up free view and you'll specify that you want to look at FS average instead of whatever your individual subject is. So FS average was created from about 40 subjects, age range, I think 50 to 90 or something like that. And this is our default registration space. So when you run Recon All and it computes the spherical morph, the registration to common space, it's registering to this FS average. And the coordinates are in MNI 305 coordinates. So at this point, your data is getting transformed into like a Talairach-like space for intersubject comparison. So the advantages of using surface-based intersubject registration are, for one thing, it's gray matter to gray matter because it's all gray matter. So you'll never be mapping and comparing gray matter to white matter. It's gyrus to gyrus and sulcus to sulcus. Some minor folding patterns won't line up. The human brain is very variable, and so some people will have some folding patterns or little bumps or gyri that other people simply will not have. And so, so in that case, FreeSurfer just tries to do the best that it can, given that it's basically a problem that can't be solved. It's fully automated, so there's no landmarking necessary. When you run Recon All, the registration to the FS average space is all done for you as part of the 6 to 18 hour process. The atlas registration is probabilistic, meaning that most variable regions get less weight. So things like frontal regions are very variable in the human brain, whereas something like calibrant sulcus or sclerotemporal gyrus or central sulcus are all, everybody has, those three. Then we have this subject FS average that is where all of the group analysis will be performed like a Talairach space. So that's intergroup registration, which is one of the two main pre-processing steps prior to doing group analysis. And the other major pre-processing step is spatial smoothing. So you smooth because you can increase the signal-to-noise ratio. And you also smooth to improve intersubject registration. So you can imagine that if whatever intersubject registration technique you're using, you had like a blob here in one subject and a blob here in another subject, and they don't quite overlap all that well. But if you smear them, if you smooth them, then you actually extend their spatial extent, and they have a little bit better overlap. Everybody wants to know, how much should I smooth? And for 15 years now, I've been having to tell them. And the reason is that it's highly dependent upon the actual test that you're doing. So it can vary. So an engineer, which I'm also an engineer, will tell you that the right size to smooth is the size of your blob. And that sounds useful until you go to realize that you don't know how big your blob is until you analyze your data, and you can't analyze your data until you smooth it. And so, so maybe you have some idea of how big you expect your blob to be, but the blobs can change across space as well. So typically in neuroimaging, what happens is that people will just kind of adopt a convention. And so typically on the surface, what we'll do is to smooth by about 10 millimeters, which is also about the same amount that you would smooth in the volume. But just so you know, it's difficult to try to establish what the appropriate smoothing level would be for your particular study and for your particular brain region. So what is smoothing? So smoothing is where you take the value at a voxel and replace it with the average of neighboring voxels. So here it is in volume-based smoothing. So if I would go through each one of these individual voxels, and I would draw a circle around it, and I would take all of the neighbors inside of that circle, and I would average them together, and I would replace that voxel by that average. And so in this case, the problem with doing volume-based smoothing is that you're mixing tissue types. So if I'm smoothing this voxel here where this plus sign is, I get gray matter that's nearby there on the cortex, but I also get this white matter, and I also get the CSF at 7 millimeters. And if I expand it to 14 millimeters, then I'm getting all that stuff, plus I'm getting the stuff that's way over here on the other side of this gyrus, of the sulcus. But people do it because, so the true distance between these 2 points is probably something like 25 millimeters if you were to follow it along the surface. And so in reality, even though you'd say maybe I have 14 millimeters here, in reality I have a much further way to go. It's more like 25 millimeters there. So in reality, like smoothing in the volume isn't such a good thing to do. So this is a slide that you don't have because I just added it last night, but I just wanted to reinforce this idea of what happens when you smooth. So this is an FMRI scan with zero smoothing, 5 millimeters, and 10 millimeters. You can see that it just gets considerably blurrier as you increase the smoothing level. And the way that smoothing is quantified is through something called the full width half maximum. And so the averaging that goes on inside of this circle is a weighted averaging. And so the further that a voxel is away from the center of the circle, the less that it's weighted. And the weighting is done using a Gaussian. And so the width of the kernel measured as a full width half max is the width of this kernel measured. So if this is the full maximum, I would go down to the half maximum and ask how wide is the kernel there. And so that's what the definition of a full half maximum is. And so the wider the kernel, the more smoothing that you get. And people smooth, again, this is not a slide that you have. People smooth for good reasons. So this is an fMRI working memory paradigm undergoing smoothing from 0 to 20 millimeters. You can see that at 0 millimeters, there's just a lot of speckle noise there, and as you increase the smoothing, suddenly you get these big blobs. But if you increase it too much, then you end up losing signal there. So this is why people smooth, because smoothing has a tremendously profound effect on your statistics. So surface-based smoothing is basically the same idea in that we're going to draw a circle around vertices and we're going to average inside of that circle. So if you remember, the surface is this mesh model. You have some number of vertices. Each vertex has some neighbors, and each vertex has an XYZ coordinate. And so instead of averaging over nearby voxels, we're averaging over nearby vertices. So on a flat map, you could just imagine drawing a circle around on this flat piece of paper and just taking all of the vertices that are inside of this circle. So if you were to look at those vertices in 3 dimensions, maybe they would, follow, hug the side of the gyrus. And when you do this, you can avoid some problems. So this is something that Bruce showed in his presentation yesterday. So this is a surface-based FMRI analysis of a working memory paradigm. You can see that you have activation here in DLPFC. You also have activation here in anterior insula. But you don't really have so much activation here in pars apicularis, right in between the two. However, when you do it in a volume-based analysis, these two blobs end up getting smeared together. So it can actually give the appearance that you have some activation where you don't really have activation there. And again, this is a slide that I threw in last night to show you a difference between surface smoothing and volume smoothing. This is a PET application looking at the density in 5-HT4 serotonin receptors between the left hemisphere and the right hemisphere. And this left image was done with surface-based analysis, the right image was done with volume-based analysis. And you can see that with the surface-based analysis, you have much more realistic looking blobs, especially as you it's changing the smoothing level from 0 out to 15. With the volume-based analysis, things are much less well organized and much harder to interpret. So finally, I'll talk about the surface-based clustering. So when you go to do correction for multiple comparisons, what you would do is to go to something like this image here in the upper left, and you would say, okay, this is a blob, which is a contiguous set of voxels or vertices that are above a particular threshold. And so when you do the clustering in 2 dimensions, you end up having a neighborhood that's in 2 dimensions instead of 3 dimensions, which means the cluster size has an area measured in square millimeters rather than cubic millimeters. And you can also improve your power, because when you do your correction for multiple comparisons, the correction severity is always related to the total amount of search space that you're looking over. Because the larger the search space, the more likely it is that you're going to find something by random chance. And so if you're looking just over the surface instead of including like a lot of white matter where you say FMRI signal, there's no FMRI signal, then you can reduce your pain for doing correction for multiple comparisons. So this is also something that Eugenio is going to talk about later on this afternoon. So to summarize why you would want to use surface-based analysis, function has surface-based organization. Function tends to follow the folding patterns. So you can get better intersubject registration by aligning the folding patterns instead of aligning simple MR intensity. Smoothing, you can smooth along the surface so that you don't include CSF and white matter or get contamination from an adjoining sulcus. You can cluster in 2 dimensions instead of 3 dimensions and correct for multiple comparisons there. And so that's going to be reduce the pain of doing correction for multiple comparisons. But in general, even though all the surface-based analysis looks more complicated, the basic principles are the same as what you would do with 3 dimensions. So use FreeSurfer. So I think we'll go straight to Eugenio.